Hello everyone, welcome to General Microbiology. So as we get started here, I want to issue something of a warning for our class. You should know going in that this class is pretty tough. It's going to be very dense with a lot of material we're going to go through. And you're going to learn a lot over the course of this class, but it's going to be a lot. It's also extra challenging because it's partly an online class. So you're going to be responsible for a lot of learning on your own time. We're going to have in-person labs every week, and then you're going to have to do a lot of extra work on your own to accomplish the additional lecture portion here, where we're going to be reading material from the book and watching recorded lectures that I've made for you each week. We're going to review some of that material in our classes, but really we're not going to have a ton of time for it because a lot of that's going to be dedicated to actual lab activities. So as you watch this video and the others for this class, I strongly advise that you do a few things. First, watch these videos in full. I know they're long. I have to record them, so I feel it. So it's important, though, that you listen to the entire thing, watch the whole thing, because it must be done. Also, strongly recommend that you take careful notes throughout. I really recommend that you handwrite your notes instead of typing them out, because there's some evidence that the act of handwriting really helps reinforce a lot of that information that you're learning, commit it better to memory than the notes that you type out. And also, this class, it has a lot of supplemental content, things that are not necessary for you to watch. They're not part of our graded material or the stuff you're going to be tested on, but it's really helpful. I've gone and picked a lot of different videos and podcasts and things like that, and I have them in folders for extra resources in each of our D2L chapters, our units. And I think that these are really good complements to the things that you're learning each week. So please don't forget about those. I really recommend you check them out. So getting started here, for millennia, many years, people have contemplated many different big questions about life. Where does it come from? What's it made of? How do our bodies actually work? What differentiates people from one another? Because we see that people are very different from one another. How do diseases work? Where do they come from? How does reproduction happen? And so much more. And a lot of these questions, they've spurned a lot of the first naturalists to study life. These are among our first scientists that have really gone to work to understand our natural world. And we do that today and back then using a framework for building knowledge. And that framework is the scientific method. The framework works like this. Essentially, you start with a question. You ask a question. And then you do background research to understand your study topic more, right? You won't have all the answers to start with. That's really why it's a question that you need to answer. So you have to have some starting point to inform what comes next. And that is making a hypothesis. This hypothesis, it's really a prediction, an educated guess. You think, it's like, what do you think is going to happen, right? What is reasonable to happen? What is a reasonable answer to your question? And what is the answer that you think is going to be the answer? From there, we have a point to go off of, a starting point. This hypothesis, we then create an experiment that we can use to test it, to see if that hypothesis is correct or not, if it is supported by the evidence or not. And what that experiment looks like, it can be a lot of different things. Really depends on the question that we're asking and what we're trying to test. We're going to do some experiments in our class that we will get some hands-on experience with this and following it through to the point where we get our results. We observe what the results of that experiment is, and then we can draw conclusions from it, right? Once we do, we can actually report that information. We can tell other people what we learned, what we found, and it can even be the springboard for new hypotheses. And it can actually wrap all the way around. If we see here that it's designed as a circle, so really the scientific method, it feeds into itself. As we make observations, we ask questions, generate hypotheses, and make predictions. We do experiments to test those hypotheses, generate results, interpret those results, draw conclusions, and then use those conclusions as new observations to fuel new questions. And along the way, we're building a lot of critical thinking skills. Critical thinking, this super important concept that is ultimately behind all good science. It's an objective analysis that we use to inform our judgment of our data, our analyses. We have to, in the process of doing critical thinking, put aside our potential biases, things that we are somewhat slanted in our beliefs. We all have these things, even though some of us have more bias than others. And we have to put aside our personal views when we are thinking about issues of science. Forget about what you want to happen. Think about instead what the data is saying. This ultimately is what we're doing with critical thinking. And so when we are applying critical thinking in a science framework like this, we're going to be asking lots of important questions, like, if my hypothesis is right, I expect to see this. What do I expect to see if my hypothesis is correct? If my hypothesis is wrong, what should I see? What do I think I will see, and why? And then after we get our results and we analyze them and start to draw conclusions from them, we need to ask other questions like, what could have gone wrong? Is there anything that did go wrong in my experiment? And if it did, how would I know? And we apply that information to critically analyze the work that we did, and also the work that other people have done. We can apply critical thinking to other people's work as well. Ultimately, this is a way for us to check our work, our conclusions, and make sure that we have reached an answer to a question that is trustable, that is backed by evidence, and has not been affected by like our personal beliefs. This whole process, scientific method, critical thinking, is so important, especially in medicine, because this is ultimately how we have built medicine, is through rigorous scientific testing, the application of scientific method, and relentless critical thinking skills, relentless application of critical thinking, and always trying to eliminate bias, our personal bias, when we are trying to understand medical issues. This all has involved following this method from start to finish, taking a step back after we've done that and reflecting on our work and potential bias and how we could avoid those things, and also constantly, relentlessly applying an objective perspective to our work. I say all this because you're going to do a lot of this in class. We're going to be doing our own testing for a lot of different research questions, including trying to evaluate an unknown species and figure out what it is, what bacteria it actually is. We're going to be applying the scientific method. We're going to be making experiments and testing these species using different methods in our classes. And I want you to apply the same rigor that any scientist would in these situations. So with that, I want to start talking about some of these early ideas that people had with some of these big questions that I mentioned earlier. Like, where does life come from? This was one of the biggest questions. And to some degree it still is, but we have much better answers for it today. Some believed early on, I'm talking in the early history of humanity. That depending on the life form that we're talking about, life could just arise spontaneously from non-living material, or dead material even. This was a concept that we called spontaneous generation. And this idea, if you looked at it in different ways, there were lots of different ideas about spontaneous generation. Some people believed that as meat rotted, it would grow and produce maggots that would turn into flies. Some people believed that if you let hay sit for too long in the field, all bundled together, it would eventually grow mice. That mice would sort of be born from these hay piles, these hay bales, which to us, it's important to note that sounds kind of crazy to our perspective, right? But it did not sound so crazy to these people. We, importantly, have our own biases, a privilege of information that we have lived in all of our lives, that we understand a lot of important info about life, that life is made of cells, that life comes from other life. This was not well understood, or not understood universally back in the day. So this idea, spontaneous generation, it became relatively popular at the time, right? This concept that living things could spontaneously arise from non-living things. Okay, so this again made sense to the people at the time. It was a reasonable conclusion based on what little they knew about the natural world. They did not understand what organisms were made of. They did not understand how reproduction really worked. So this hypothesis, this idea that they had, was based off of observation at the time. People would see that maggots would seemingly arise from rotting meat if you left it long enough. But people weren't sitting around to watch the meat rot. They wouldn't see flies land on the meat and lay eggs on it. The eggs were too small to notice with the naked eye. Flies would come to feed on the meat, and they would leave, and then later maggots would arise, and that flies would then arise from the maggots. It was believed at the time that those things were all connected in a very linear way, that meat rots, turns into maggots, and then the maggots turn into flies. There were similar beliefs, lots of other connections, like, for example, molding bread. When bread would mold, you wouldn't see the spores of these fungi land on the bread and then slowly take it over to make it mold. That was something that happened on a scale that was too small for us to notice. Similarly, when you would see flies go through their life cycle, this took too long. It was too slow for us to notice. And so people didn't. They simply did not notice these things. And so people were left instead to make other hypotheses, like the ones we mentioned. And we've kind of always grappled with the impacts of microbial life, things that are too small for us to notice, without even really realizing it. So this makes a lot of sense to us. People suffer from disease. People have always suffered from disease. But disease is caused, or infectious disease at least, is caused by microorganisms. And we've been treating disease for as long as we've been getting disease. And all this time, we've never, up until very recently, like shockingly recently, we have not noticed what causes disease. We'll talk about that more in a little bit. Similarly, like food, the fermentation of food or the spoilage of food, these types of things, we knew that they happened, not how or why. So, like I said, we've not understood for the longest time what was causing a lot of these changes in our environment. But that started to change when the first microscopes were created. So these microscopes, they date all the way back to 1585, where you have the first compound microscope that was developed. And these microscopes, they were made kind of on accident. The people who were making these were not interested in microbial life or looking at things on incredibly small scales. They were astronomers, and they were interested in space. So they were trying to make telescopes and were dabbling with different lenses and pairing them together. And in the process, they created the first microscopes as well. So later on, about 30, 40 years later, Galileo, he also developed a microscope that he, it had multiple lenses. It was a compound scope. He never even bothered to name it because he couldn't give a crap about it. He was an astronomer first. And so, it was later in 1625 when other people who were started to take an interest in some of the things that Galileo had and made, that they started to call it microscopio. So these microscopes, as they were developed, they were, really pioneering the discovery of microorganisms because, again, before that, people could only speculate on the existence of life too small to witness. A man named Antoine van Leeuwenhoek, he's sort of viewed as the father of microbiology. That he created a lot of his own microscopes. This design over here, it was kind of interesting. It was a design that was he produced a lot of, but was rediscovered in 1957. So we didn't even know that it existed until then. And Leeuwenhoek was fascinated with the microbial, and he would put just about everything under his microscope. Essentially, this little spot right here at number 1, it marks a little pinhead that you would place small samples on, and you would hold this paddle up to the sun and look through this little pinhole on this side over here down at the pinhead, and you'd be able to see a very magnified version of what you're looking at. It's a really amazing tool. He would put all kinds of stuff: water samples, saliva, plaque from his teeth, fossils, tiny pieces of plants and animals, all kinds of stuff. And he would make a lot of his observations. It was very cool. Here on the right, we have some of his drawings, some of the stuff that he was seeing. So a lot of these are very likely bacteria that he was drawing. Very simple shape, varying in shape. Some of them are twisty, some of them are little dots, some of them are long rods. And he notes here that some of them can even move on their own. So this was very notable. And so a lot of his drawings are like this. Also, I like to show students these drawings because I love to draw observations, and I always strongly encourage students to draw what they're observing, especially in a microscope. And they don't have to be works of art. They can look like, kind of crappy, as long as they communicate what it is that you're seeing. That's all that matters. So take it from Leeuwenhoek here. Leeuwenhoek called these life forms animalcules at the time. It was a catch-all term for anything microbial. All living things that were microbial, everything from bacteria to small animals and plants to little amoebas and things like that. So the concept of this, though, it's not something that I think that Leeuwenhoek came up with himself. The concept of animalcules was not something that originated with him, but he took that word to refer to, anything that was too small for the naked eye that he was seeing in his scope, right? And so the concept, the term is definitely older, right? So this is a quote from Marcus Varro, 30 BC, and it says, "Note also, if there be any swampy ground, both for the reasons given above and because certain minute animals invisible to the eye breed there and borne by the air reach the inside of the body by way of the mouth and nose and causes disease which are difficult to be rid of. This is actually pretty profound because this is a guy who is speaking about infectious disease caused by microorganisms, or these little animalcules, before people realized and could demonstrate and prove that diseases were caused by these things by many centuries, many centuries. So with the advent of the microscope, a new line of thought emerged. So the question really became, what does this mean for other living things like us? Now that we could see all these tiny things that were so small that we couldn't see them with a naked eye and only through this scope, what does that mean for other living things? Well, Robert Hooke was really interested in this question, and he started to investigate this as well. He was a very early explorer of the microscopic world, and he is the person who first described the cell. He did this by looking at a couple different species, mostly a fungus called mucor. That's the genus. And he would also look at slices, very thin slices of cork. And he would see that in both these groups, he would see little containers inside. He called them cells. He named them after these living quarters that you'd find in monasteries, like this one, where each and every one of these little spaces here, they were little living quarters. There's a door back there, and they're very small, confined, very restricted. And so he looked at these, and that's what he immediately thought of, was the cell. And so, as Robert Hooke investigated this material, he started to notice that just about everything he looked at, every living thing that he put under the microscope, had cells in it. It was made of cells. And interestingly, Hooke did often communicate with Van Leeuwenhoek about this observation. And so together they did a little bit of correspondence about this. Everything seemed to be made of cells, according to Hooke. At least one cell at minimum. And this idea, everything alive seemed to be made of cells, was a consistent observation from then on. People who continued on this research over the next 200 years would continue to make that observation again and again and again. And it just, no matter what they looked at, no matter what living thing they looked at, whether it was a plant or an animal or a fungus or a bacterium or anything, they were all made of cells. It took over 200 years, though, for us to kind of reach that final conclusion where we were pretty confident. Like, yeah, everything alive is made of cells. And so two major players in this game was Theodor Schwann and Matthias Schleiden. These were animal and plant biologists, respectively, that were asking these same questions in their respective groups. So Schwann was looking at animals and looking at like tissue samples from various animals and finding over and over and over again, yeah, cells. And Schleiden was seeing the same thing in all of his plants. And so this, they put a lot of literature forward that recognized, yeah, this stuff, life is made of cells. Their respective groups are. And their assertions built on a lot of hypotheses that were being constructed before them by previous scientists. There was a slow building realization that maybe spontaneous generation was not right. This was a prevailing idea for centuries upon centuries. And it was just kind of accepted by a lot of people that this is just the way that it is. Life can arise from non-life. It can just spontaneously arise. But the discovery of microscopic life and the discovery that life is made of cells, this information starting to poke holes a little bit in this idea, because the idea now arises: what if life is arising from life, but it's happening at scales that we can't see? So if you were a scientist back then, think about this. How would you test this idea of spontaneous generation? Take a moment right now to think about what kind of experiment you could run. Because a lot of people have had these thoughts, and a lot of people have made these experiments and applied them, tested this idea of spontaneous generation. We're going to talk about that, but again, just a moment. Think about it, and let's go on. Pause if you need to think about it more. So this is a man named Francesco Redi, and you're going to hear about him occasionally throughout our class, particularly in the early chapters. He was among the first to assert that life only came from life. He said, in some of his writing, he said, omni vivum ex ovo. Excuse my bastardization of that Latin. But it says that every living thing from an egg. So his assertion was that all living things ultimately arose from something incredibly small, an egg that is too small to be seen, or not seen. And so he made an experiment in 1668 where he left meat out to spoil, and he had two test groups. One of them was a piece of meat that was left out in the open, exposed to the elements, exposed to the environment. One was left sealed and covered in gauze. And so the flies, what he found was that the flies only ever lived in the open container, the container that had the meat left out into the open. And only maggots arose on that piece of meat. So his first experiment was actually like this. It was this group over here, where it was one piece of meat out in the open, one piece of meat fully sealed away, right? And what he found was that only this piece of meat grew maggots, or developed maggots and flies. The other one did neither. It didn't get either of these things. And researchers at the time thought, or other people are speculating at the time, thought that, well, maybe there's something going on where it can't access the air, and so the living things inside died, like as they were born, or something like that. Like there were various like naysayers that came to Redi and said, well, your experiment is flawed. So he made a revised version where he introduced a new test where he left a piece of meat out in a container that was covered with gauze. And so it was breathable. And what he found was that maggots and flies actually landed or grew or were sort of born on the surface of this gauze and not on the meat itself, which indicated very strongly that they were not coming from the gauze or from the meat, but instead they were attracted to the meat. So this was a pretty effective and profound experiment. There were several other people over the next couple centuries that did similar experiments. We're not going to cover these in huge detail, but they explored a lot of this stuff. So we had Pier Antonio Michelli in 1729. He was working with fungi that, looking at how fungi colonized the like fruits. So he actually would spread and place fungal spores on exposed pieces of melon, and was able to demonstrate that fungi were born from spores and not from spontaneous generation. They didn't just arise from food as it rotted. Lazzaro Spallanzani, he is from the 1760s. He did a really cool experiment where he boiled 2 flasks of like beef broth. And again, very similar to Francesco Redi's experiments. One of them was left open and the other one was corked after boiling, and only one of them became rancid. Only the one that was exposed to microbes in the environment. Despite these experiments being quite convincing, the idea of spontaneous generation still stuck around for a long time. Even people that were pretty profound in this early cell bio field had their own kind of kooky ideas. So Theodor Schwann, who again was one of these people that was actively putting forward research that said that life was made of cells, that all life descended from cells and was made of cells. He recognized this from his research, but he also believed that cells themselves could spontaneously generate. This idea, what he called cell crystallization. So he asserted that you could have spontaneous generation or formation of cells from non-living things that could eventually become a whole organism. So some people had some pretty interesting ideas at this time. It was another researcher named Rudolf Virchow who ultimately ended up convincing Schwann that cells could not arise spontaneously, that they had to arise from other cells. And Virchow, he did a lot of, he talked a lot about like the generation of cells in during embryogenesis, like when an embryo is forming. So he looked a lot at chicken embryos to do that, and so this was among one of the earliest recognitions of how cells actually divide, how they do mitosis to form new cells. Except that Virchow didn't really do that work. He actually stole that from another scientist at the time, a man named Robert Remak, who was a contemporary of his. It's kind of interesting because it kind of has to do with a lot of prejudice at this time. That Remak was Jewish, and it was sort of like a thing where Virchow felt acceptable to steal from this person because he was not of the same religion and things like that. So it's kind of interesting that Virchow ended up publishing his Remak's work as his own and got a lot of credit for it up until very recently when we figured out, oh, he stole that. So in the end, a final theory was assembled from this collective body of work that is known as cell theory. And this is 100% one of the most important things I want you to know and be able to explain, okay, and talk about. Cell theory, it has 3 tenets. The first one is that all life is made of cells. All living things, all forms of life that we know are constructed and built of cells. Not other stuff, but of cells. Number 2, the cell is the smallest unit of the organization of life, right? Any kind of organizational unit that is smaller than a cell is not living. And number 3, cells can only arise from pre-existing cells. So all living things are descended from previous living things, because those cells that made up that living thing had to replicate to create more cells to make that new organism. This last one is very profound because this also means that all living things have this long, unbroken chain of relationship, a descent through history, that all living things, no matter who we're talking about or what, are related to one another, either very closely related, like other humans are to one another, or much more distantly related, like humans are to, wombats, or if we keep on going further back, fishes, like trees, flowering plants, even bacteria. We are related to all of that life because all of life has descended from previously existing life. So it's more akin to view us all as cousins of one another, with some being further removed than others. So Schwann and Schleiden, they both believed that this only applied to plants and animals at first, but soon they recognized and we collectively recognized that this was all life, and not just complex multicellular life like we are, but also single-celled organisms like bacteria and protozoans, different things like that. So this leads to some interesting questions, though, because we can also ask, if a thing is not constructed of cells, then what is it? Well, the simple answer to that is that it's not alive. So this includes viruses, prions, rocks, minerals, water, gas. These things are not alive because they are not made of cells. Even if they behave very much like a living thing, like viruses. Viruses are so perplexing because they can reproduce in that they kind of hijack a cell to make more of themselves. They can evolve like a living thing does. They behave in some ways like a living thing, but not others. And most importantly, they are not made of a cell. They don't have a proper cell. And so even though they feel in some weird way like they're alive, we don't classify them as alive, although this is at times subject to debate among scientists. Suffice to say, in my class, we're not going to treat them as alive. So let's continue on with spontaneous generation, because as cell theory arose and became more and more popular, there were still these hardcore adherents to the spontaneous generation idea, this old theory. And so this really culminated in the 1850s when a man named Louis Pasteur finally kind of put this stuff to bed. So in 1859, Louis Pasteur did a very similar experiment to Spallanzani, where he had this really cool, called a swan neck flask, where it has this long swan-like neck, and this neck, essentially the length of this neck, it makes it hard for bacteria to move all the way through this area. And you could do something really interesting too, where if you had fluid in this neck and then you heated the fluid up, the fluid would evaporate, some of it would, and it would start to move through the neck, but the cool glass at the far end of the neck would cause the liquid to condense and turn back into a liquid from a gas. So you would end up with this thin layer of liquid here that would, in effect, seal off this part of the tube from this part of the tube. So bacteria, yeah, they could come in here and they could go into this part of the tube, but they could not make their way back up through the tube after it's been heated. And so Pasteur made these two flasks where he put broth, like beef broth, inside these swan neck flasks. He heated them up because, keep in mind, at this time, Pasteur has observed bacteria at this time. He has used a microscope. He has observed living things smaller than the eye can see, and he believes that they are the cause of food spoilage, not like some chemical process, not this liquid turning into living things and then spawning stuff from it. He believes, because he has seen things like yeast, things like bacteria, he has witnessed these things with his own eyes, thanks to a microscope, that he believes that they are causing food spoilage. So his method looks like this. He heated these two flasks up, but one of them, he snapped the neck off to expose the broth to the environment. And he left them both for a set amount of time out and exposed to like their environmental conditions. And what he found was that the broth that was heat sterilized, that did not have the neck broken, grew nothing in it. It was still fresh. quote fresh. It was not contaminated by bacteria, but the flask that did have the neck broken off, it was contaminated, and it did become fetid. It became rancid because bacteria grew inside of it. And you could actually take the material out of here. You could take the broth, put it on a slide, and look at it and see microorganisms that were in it that were not in this flask. So this was pretty profound. Today, we have come to know that living things can be incredibly simple. They can be single-celled organisms. They can be microscopic, too small for our eyes to see. And so, as we have learned that, new questions begin to arise about these microscopic forms of life. Why are they so small? What is their relationship to us? Can microorganisms develop into macroorganisms? And what exactly do they do anyway? What are they doing in their environment? And so we've come to a lot of these answers, but, at the time, this was such a new science that people were still trying to figure this out. A lot of this stuff, it started to pave way for new ideas. So one major idea that was prevailing at the time, before the onset of Pasteur's research, it was miasma theory. It was this idea that disease was ultimately caused by bad air. That you were exposed to foul, nasty winds, stagnant, bad-smelling air, and it would disturb something in your body and cause an illness. And specific smells like rot and sickness were attributed to this in a higher degree. And so the idea was that you could avoid disease by avoiding bad air, right? This idea, it made sense to people at the time, and there was certainly more supporting evidence for this idea than there was for spontaneous generation. I mean, people who would live around really foul-smelling places, they were often prone to sickness. People that lived near like cemeteries or like in like sewage, different things like that, you would expose yourself to more pathogens. Not that they understood this at the time, but that would cause a lot of illness. You'd be more prone to it, certainly. And then you also had the experience where terrible smells would also make you feel sick. Certainly, we've experienced that before. You smell something really bad, and it makes you want to vomit. And there's also the association that sometimes sick people could smell sick. People that are suffering from a bacterial infection, that have like an infected wound, that wound, because the bacteria smells, that wound smells. And so it can be really stark and powerful, pretty nasty. This is incidentally is why the classic plague doctor like outfits and everything, they had that big beak. They filled that beak with like flowers, potpourri, things that smelled really good because it was their way of fighting the sick smells while also treating patients. They would go and actually see people who had these infected wounds. When the Black Plague was going on, they would go and see people with like these infected buboes. So they called the wounds that would develop from somebody that had the Black Plague. And they would be able to treat them while still avoiding this stinky, nasty, bad air that could make them sick, so they thought. Of course, we know today that miasma theory is incorrect. We know that bacteria and viruses are what cause disease, that are like microorganisms, really. So bacteria, viruses, fungi, different protozoans, even some animals can cause disease. And it's infectious. And so ultimately, what people who believed in miasma theory were seeing was, interestingly, a relationship between microbial activity, and the bad smells that they used as evidence to support their ideas. So when we discovered microorganisms, that really changed the way that we saw a lot of stuff, particularly disease. So again, we have to thank Louis Pasteur for recognizing that microorganisms, germs as we call them, cause disease. This idea really originated with him, that he is a major player in this game, that when he disproved spontaneous generation, it was not long after that he asserted that germs were responsible for infectious disease. And later, a close rival of his named Robert Koch was a German who was, I guess the Germans don't like the French so much. They were at odds. They were at each other's throats all the time. They worked not together, but opposed to one another, pushing a lot of amazing research forward. And Robert Koch was responsible for figuring out some of the first methods to demonstrate the causative agent of a disease. So those things are called Koch's postulates. We still use them in some form today, and we can use them to demonstrate that a disease is caused by a pathogen. For example, in recent time we had the COVID outbreak, and we were able to use, it was not exactly as Koch described, but we used a modified Koch's postulate to figure out that COVID-19 is caused by a specific virus called SARS-CoV-2. And so we were able to identify that virus, name it, and start designing methods to effectively treat it and prevent it. Importantly, microbes are not all these villainous actors that are out there causing disease all the time. Some really important work was done by people named Sergei Winogradsky and Martinus Bezernik. That these are two Russian researchers, that they realized that microorganisms were present just in the soil, and they were doing things totally separate from us, not causing disease, but actually doing some really important jobs in our environment. They were responsible for this concept that we'll talk later on in the semester called nutrient cycling. It's the movement of nutrients from one organism to the next, or from one part of the environment to the next. So it comes in a lot of different forms. It might look like one organism eating another, or feeding on another, stealing nutrients from one another. It might look like decomposition, where an organism's body after they die, or maybe their waste after they have relieved themselves, that other organisms can make use of that and can break it down into smaller, simpler resources and then use it to make things with, to build new cells with. And even drawing nutrients from inorganic sources, turning them into organic sources. This was a major revelation, because up until this point, in that brief window between the discovery of bacteria, the recognition of germ theory, and this point, right? People believed that bacteria were just exclusively agents of disease, and that they were not doing important things in our environment, but in fact they are. We call them free-living organisms. They live freely in the environment, not as parasites, but as grazers, as decomposers, maybe even as predators of other things. And they serve these really essential roles in cycling important nutrients, moving it throughout our environment. Today we recognize that microorganisms are really important for their environment. They have their own ecology. That means the way that they interact in complex ways with other forms of life and with their environment. That includes other microscopic organisms and even macroscopic organisms like us. The reality is that even for those infectious bacteria, the ones that cause us disease, we are also part of their ecology. They interact with us, but as parasites. there are plenty of other organisms that interact with us not as parasites. And in fact, these organisms are so influential, they shape their environment in really substantial ways. We're going to talk about some of those later in the semester, but it's pretty profound, the way that they have shaped our planet over the millions and millions and millions, really billions of years that they've existed. And so we can thank these organisms for the fact that we have a habitable, oxygen-rich atmosphere, that we have healthy nutrient cycling on our planet, that we have sources of oxygen from photosynthesis that are being actively done all the time by lots of different microbes. And in fact, those microbes are often much more impactful on the production of oxygen than large plants like trees are. So they're really influential. And of course, we are impacted very directly, not just by them causing disease for us, but also a lot of these organisms live on or inside our bodies as partners with us. So our bodies are actually home to a huge, diverse community of microorganisms. Billions and billions of non-human cells that live in or on your body. Would it surprise you to learn that more than half of your cells that make up your body here are not human cells, but bacterial cells? Almost half, if not a little more, are made of they're bacterial or fungal or archaeal. They're not Homo sapien cells. They're not human cells. Now, by sheer mass, by our body weight, that's only 0.2% of our collective body weight. So maybe a few pounds worth of bacterial material are in your body at any given time. But it's still such a startling number. They outnumber us in our own body. But it's a good thing that they do. Because these organisms, they are mutualists. They are partners with us. They live in or on our body and provide a lot of value. They help us digest our food. They protect us from disease. This community we call our microbiome. And so in our gut, for example, we can give it additional names. We call it often our gut flora. And flora in this case is just a relic term from this old time that we used to think that bacteria were a type of plant. We now know that they're their own group, but some of those names still stick around. So this microbiome, our gut flora especially, is very important for our health. So healthy people are thoroughly colonized by microorganisms. I apologize that this makes you a little bit squeamish. I hope it doesn't, but it's just the reality of things. Mostly bacteria, with some fungi and occasionally some animals, some very, very microscopic animals. And most of these organisms are totally innocuous, even friendly. But under some different circumstances, some of them can cause disease. Not all, but some. So we should talk about some of those things. Pathogens. Okay? Pathogens are any kind of disease-causing microorganism. They cause disease in a healthy individual. We can have normal pathogens, like typical pathogens. These are pathogens that their specialty is causing disease. That's the way that they choose to live their life, the way that they've evolved to live their life. Opportunistic pathogens are, as the name indicates, they're opportunistic. They don't usually cause disease, but they could under the right conditions. If, for example, you had an injury and bacteria that lived naturally on the surface of your skin was pushed through the layers of your skin into the fascia of your body, right? Down into the dermis and beyond. That is an unusual situation. The bacterium is not where it normally grows. It's been placed into a different environment. And so that can cause it to suddenly have to sort of change lifestyles, and it becomes a pathogen as it grows, it causes problems for us, causes disease. Alternatively, maybe you have a situation where your immune system starts to falter. And normally, your body would have no issue keeping a lot of this stuff in check. There's a lot of back and forth that happens with organisms that live in our body. If they end up in a place they shouldn't be, or they start doing something they shouldn't do, our immune system comes in and kind of hits them, brings a smackdown on them, and puts them kind of back in their place. And says, no, you can't cause disease. It's a great example of what happens with Candida albicans. Lives naturally in a lot of the surfaces of our body, particularly in our orifices, our mouth, our genitals. And so if things are kind of out of balance, like our environment in those orifices is not right, or our immune system starts to falter, Candida albicans, every once in a while it tests the boundaries, starts to grow like a pathogen. And if our body isn't there to smack it back into place, it becomes a problem. That's how we get a yeast infection, or we get thrush, or various other types of similar illnesses, right? So those are opportunistic pathogens. Not typically pathogens living in our body. Usually they're there doing relatively friendly things, but they can, under the right circumstances, cause disease. In our bodies, we have a lot of different defenses that keep opportunistic pathogens at bay. Most of our body is kept very sterile. So we have a lot of orifices inside of our body. We have, our mouth, our sinuses, our throat, our lungs, our digestive system, and our genitals as well. That are spaces inside of our body that are loaded with bacteria, but there are lots of places in our body, like our brain, our spinal cord, our bones, our muscles, the connective tissue, the lower layers of our skin, our dermis and epidermis, and some of these organs like our liver, our kidneys, different things like that, where they are devoid of non-human cells in a healthy individual anyway. If you do have bacteria living there, that's a problem. Now you're talking about you have like sepsis. Or like potentially a massive like skin infection or organ infection, or maybe you have meningitis, or different things like that. So we spend a lot of effort, our immune system does, keeping these parts of our body free of microbes. And when these opportunistic organisms kind of get out of line, like I said, we have immune responses to stop that, usually before it becomes a problem. But interestingly, here's the super cool part about why our microbiome is so cool. Those organisms that live inside of us, a lot of them are so friendly, they will actually fight for us against potential invaders. So other microorganisms that would cause us harm, if they show up in that environment, they have to, if they're going to survive in that environment, they have to get past our friendly microbiome first. And that friendly microbiome, they are in competition with those new intruders. They want to keep your body free of those intruders, because if those intruders show up, they're going to have a hard time. They're going to have a hard time accessing the nutrients that you provide them with your food. They're going to have a hard time sticking around and keeping control over this space they're living on, all that stuff. So they compete with these invaders. And because they've been long established, they're quite, a good match for you. They do a pretty good job of defending your body from these invaders most of the time. So, diseases that are caused by pathogenic microbes are typically infectious, or they can be if they aren't normally. And this really just refers to them being capable of being passed on to other organisms, and so that they develop the same disease. And even opportunistic pathogens can be infectious. A great example of an opportunistic pathogen is like a staph infection. It's caused by a bacterium called Staphylococcus aureus. And staph infections are certainly infectious. Even though that microbe grows naturally on our skin all the time, it is still very possible to contract an infection through a staph infection. A concept called emerging disease I want to talk about. This is when you have a novel disease, like a new disease, freshly emerged, that is increasing in incidence, becoming more common. For example, this E. coli strain here, right? This E. coli 0157H7. This is a new strain or a new variant of this species, Escherichia coli, that has evolved to be toxin producing. It can make a toxin that is dangerous for people. And even though normal E. coli can grow naturally inside of our bodies, in our guts, it does not normally cause disease in that scenario. If that E. coli is this particular strain, it can. So that creates a major problem, right? This 0157H7 strain. This is what causes severe disease in people. It can kill. It's pretty dangerous. And it's a new strain. It recently evolved. It recently emerged, which makes it an emerging disease. I think that we can think of lots of other emerging diseases right now. COVID was recently an emerging disease. It emerged in 2019 and became particularly problematic in 2020, where it spread everywhere. So that was a very profound emerging disease. But we battle new emerging diseases all the time. A lot of them don't become that much of a problem, but the ones that do, we know them. Then we have re-emerging diseases. These, on the other hand, are familiar diseases that are regaining popularity, much to our disappointment. Their incidence is increasing. These are diseases that were once abundant and then became quite rare and are now becoming abundant again. Old species, familiar species that are becoming a new problem. A lot of familiar and noteworthy pathogens we should talk about include measles, pertussis, and tuberculosis. But there's lots of others, a lot of others. And so some of the most current cases, like measles and pertussis, these are diseases, they're viral diseases that are, we have vaccines for them. And because of aggressive vaccination, that we have been very good throughout our history, when these vaccines were first created, to vaccinate our whole population, a huge percentage of it. We were able to reduce the incidence of these diseases to a few hundred to a few thousand every year, right, in our country. And in some cases, we actually went quite a long time without any measles cases. That we reduced pertussis quite a lot to being very reduced. And now today we're seeing a lot of vaccine hesitance, a lot of vaccine skepticism, and new anti-vax ideas that are being pushed by a lot of different people. Unfortunately, a lot of those people are both in popular culture and also in politics. It's been very frustrating to watch these diseases become re-emerging. And now, at least as I'm recording this now, there's an ongoing measles outbreak in parts of the United States. And pertussis still happens in large quantities of people. Unfortunately, these diseases are most impactful for children that are not old enough to get their measles or pertussis vaccines. And so they're the greatest casualties. They have the highest rates of mortality and also the greatest impact if they get infected. And so today, unfortunately, these diseases keep going, and they propagate particularly among people that have not been vaccinated. Lesson here: if you haven't gotten your vaccine, think about it. You really should do that, because you can mitigate your risk of getting measles or pertussis or a lot of these other diseases like mumps, rubella, tuberculosis. Sorry, not I don't know if we have a vaccine for tuberculosis. A lot of other ones, right? And in particular, you mitigate that risk of passing it on to someone else who might be more at risk than you. So, pretty impactful. Tuberculosis is becoming a new problem for a totally different reason. It's evolving antibiotic resistance. So fun. Well, that's a new problem that we have to deal with, and it's getting to the point now where this antibiotic-resistant tuberculosis is spreading and becoming very difficult for us to treat and deal with. It's not impossible, but it's much more challenging. This is the crazy thing about this disease. It is a disease that still today has an incredibly high incidence and mortality. Like the number of people who have died of measles is really high, and it is curable with antibiotics. But now we have these additional problems, these new complications, where these strains are emerging that are antibiotic resistant. This is a major concern for us in the medical industry, the medical field, that both in people who are making new medications, who are treating patients very directly, who are doing this research or applying it in the field. That this is really important, really impactful, and it's worth noting because these diseases are infectious, they are capable of spreading to anyone, who is not immune to these things. An infection or an outbreak anywhere is a problem everywhere. So it's important that we respect that we understand that, and we keep that in mind as we're treating patients and as we're dealing with vaccinations. So the last thing that I want to say on this is to go back to something I said earlier, and that is the relationship between living things. That final tenet of cell theory, this idea that all cells are descended from pre-existing cells. It's a link that links up all living things together. Everything. Us, your pets, the trees in your backyard, the fungi that are growing on the bread that you never threw out, and you probably should have thrown it out already, even the bacteria in your gut. All of those things are related to one another, genetically, directly related. That we're all effectively cousins of one another. All these living things are related, and so we share this relationship, this common ancestry. If we go far back enough, that all of life descends from this shared common ancestor, and we can learn so much about ourselves because of this by studying the qualities of other living things. Because if we understand other living things in some way, we're understanding ourselves, the history of evolution of these forms of life, these core biological processes that keep them going. Homeostasis, the process of maintaining livable conditions inside of a cell and how that works. What DNA is, how it works, how genetics are inherited, and so much more. This is a very rough diagram that represents our relationship to living things. So all of life falls into three major categories. We call them domains. Okay? We have bacteria over here as its own major group. Another group called archaea, very similar in appearance to bacteria, but very distinct as well. And then we have eukarya. And in eukarya, we have all of these more complicated things that have nuclei, things that have organelles, and we have very familiar groups like slime molds and ciliates and plants, fungi, animals. Yeah, I know that some of these things probably not so familiar to you, but we're going to talk about some of them in some of our units here. And so this tree that we're showing here, or maybe more of a bush, shows that all of these living things, they all connect at some point. Their ancestors descended they reproduced, and their descendants became all of these forms of life. So this is really important to keep in mind. So all things, we're all connected, right? Shared evolutionary history, and we can actually trace our lineage and the lineage of all these groups back to a shared common ancestor, some 3.7 billion years ago, and maybe even older. It's important to keep in mind, again, because as we learn about these bacteria and fungi and things like that, we're not just learning about these things and the way that they infect us and stuff. We're also going to be learning in some ways about the way that our cells work, the way that our bodies work, the way that our DNA works, because these things are directly related to one another. In any case, this marks the end of our first unit. Please make sure that you are up on our next units as well, that you're watching them as they come up in our schedule. Don't miss any, please. It's also important that you go and look through the first chapter, the corresponding chapters of our textbook as you are reviewing this information, because there are some things that are in my slides that are not in the textbook. There are some things that are in the textbook that are not in my slides. Okay? All right. I will see you in class.